the eukaryotic gene expression is a little bit complicated, not like the prokaryotic like bacteria, the lack of their own gene expression. So we can a little bit understand the process. It's again the DNA molecule, if it is like this, so it has again specific region. This region is called the gene. And before this gene, there is other regions. So well-known region is called the promoter region. So what is the function of promoter region? Now, if we understand gene expression from the previous one, let's assume this one is a cell, right? And then this cell need the chemical stimulus. So chemical stimulus is coming from outside. So when the chemical stimulus brings the signal to the cell, and the chemical stimulus should pass signal to the nucleus. So then there is a pathway to that, and generally we refer this as signaling, chemical signaling. So chemical signaling involves a lot of other chemicals inside the cell. So first thing is, there are many different kind of chemicals collectively known as transcription factors. So transcription factors bind to the promoter region. So when the transcription factors, I just abbreviated TF, transcription factors bind to the promoter region, it provides the right shape and structure for RNA polymerase to bind. So now this is the time for RNA polymerase to bind. Then otherwise it's difficult for RNA polymerase to bind. So transcription factors are specific to this region, and they can first go and bind, and then which can activate RNA polymerase to bind. And sometimes then this can be further regulated by the bending of this DNA molecule, and then another region of DNA molecules, like this bending, another region of DNA molecule, they also interact with this promoter region, right? That's called the enhancer. So now you see number one is transcription factor, then number two is RNA polymerase, and number three is this enhancer. After binding this DNA, binding proteins bind to this region, bending proteins, then it interact this transcription factor, RNA polymerase and enhancer. Finally, this RNA polymerase can start synthesizing messenger RNA. Then the mRNA is synthesized, then the protein is synthesized. So can you copy this down another eukaryotic gene expression regulation? First one, this region is the gene, and it has the regulatory regions. The regulatory region, the most important thing is the promoter. The promoter is this region. And transcription factors, RNA polymerase, and the enhancer interact at the promoter region. Then RNA polymerase can move forward to make the messenger RNA. So TF indicate transcription factors. Let's see, transcription factors are kind of a proteins, small protein molecules, so they can bind to the DNA molecule. Help RNA polymerase bind to the promoter region, and after that the enhancer can further interact, then RNA polymerase activate. So app for transcription. That's translation. Done. Okay, if you see the text written under this, so it's basically the eukaryotic gene expression is regulation of transcription, and RNA polymerase binding the promoter regions, which is facilitated by the transcription factors. And then last one, sixth point, it says the enhancer bind to the DNA promoter region. So then RNA polymerase can start the transcription of the gene. Okay, done. Right now, another important thing we should understand if there are um, if there are 20 to 25,000 genes, then we can have some idea that then it can produce only 20 to 25,000 proteins. The human gene consists of 20 to 25,000 genes, and only human can make that much of proteins. But when we are trying to learn about the protein molecules, if the protein molecules, let's say one protein molecule, these are the few amino acids like let's say A, B and C, 3 amino acids. And there are many other amino acids. You can see these two directions because protein is made out of many amino acids. And if the same type of proteins, only this region change to another 3 amino acid, let's say X, Y, Z, right? So this is the protein molecule, and this one is also protein molecule. You can see two protein molecules. Only the changes we can see here and here. Just 3 amino acid change at this region, and then 3 amino acid change at this region. Otherwise, the amino acid number and the order is same, from the other two big regions. Then the question is there a separate gene for this one, and is there a separate gene for this one? Why we are asking this question? We can define, okay, human has 20,000 25,000 protein. Let's take one example, one kind of proteins. We call it antibody. I think even though A-level we haven't learned yet this immunology, immunity, you know what is antibody. Antibody is important for the immune system. Again, antibody is a protein, Y-shaped protein molecule. Okay, then we can make a conclusion, right? There is a gene. This gene can make this protein. And again, we come to another conclusion. There are 5 different antibodies, types of antibodies, right? Let's say example like this. So there are 5 different antibodies. Then we can still conclude, okay, then still fine. There are 5 genes which can make these 5 different type of antibody. Now we are going a little bit more towards now. If we look the specific region responsible for identification of pathogen, right? Let's say this is a kind of a pathogen, and this is another kind of pathogens. So this is another kind of pathogen, and that's let's say like a virus-like thing. So if we trying to understand this, maybe we can make a list like a millions of different pathogens. So if we have a millions different pathogens, they have differences. They all have some differences. They are not same. Then how can only 5 types of antibody can identify this different region? Because let's say if they express a protein shape like this, now this pathogen has a protein, it's shaped like this. Then this antibody should also have a region which can fit into this yellow triangle shape, like this. Example like this. Now this antibody specific to identify this pathogen. The other pathogen has this kind of identification marker. The other pathogen has a different identification marker. Then you have an idea, okay, then the antibody should have a specific shape on this region that can identify different pathogens. Then the question is there different genes to make different antibodies? If it is the case, we make a conclusion, let's say millions of pathogens, different pathogens, and millions of different antibodies. If we have millions of different antibodies, is there millions of different genes? And we said we have 20 to 25,000 genes. We don't have millions of different type of genes. But I gave one example like this, but we can find many examples. This is a very good example because in the future you will understand our immune system has antibodies. They can recognize pathogens which are not yet born. Example, coronavirus, COVID-19, 2019-20 around, 2019 around came. That's a new virus. But human body has antibodies to identify coronavirus, the COVID-19 virus. So we have antibodies that can identify the pathogen which will come in the future, because they have a specific shape at this region. If they have a specific region, then this protein has specific amino acid at this region. Then they can make only this shape. Because if you look the protein lesson, different amino acid has different R groups. Then the R groups fold. When the amino acid folding, R group make different bond. Then they make different shape. There should be then different amino acid and different order, right? So this is one example. We need to find answer. So you understand there are proteins, but there are proteins within the proteins. There are small differences. Such millions of differences can be made by different genes or single gene. So we're going to find answer for this question right in the next lesson. So this lesson is called RNA splicing. So precisely, let's say mRNA splicing. Okay, just to understand mRNA splicing, we again trying to see the structure of the DNA molecule. So when we look at the DNA molecules, we can divide DNA molecule into different regions. Different region are like this. I will mark here like this. Example, these three regions we can place into one group, the shaded one. The shaded three region we can place into one group, and the not shaded region we can put into another group. So these two groups are named as exon and introns. Let's say this one is exon, and this one is also exon, and this one is an exon, and these regions are intron. So then basically we can find exon and intron region of the DNA. At the moment we make small conclusions. Introns do not code. Do not give codes to make proteins. So only the exon make the codes. Exon make codes to make the protein. And you will see in the future what is the function of actually intron. If they do not give codes, and if they do not make proteins, what is the function of introns? You will see we have to still know a lot of things about DNA molecule. DNA make codes to make proteins, but they have other function as well. Right now, during the splicing process, what is going to happen, this complete sequence, let's say this is called the gene now. So from this end, A to the B is a region of gene. Now this is copied to the messenger RNA by transcription. Now this is a messenger RNA. So messenger RNA molecule also has now the same region. And the length of the messenger RNA is similar to the length of this gene, right? So this is called pre-mRNA now. Now after pre-mRNA is formed, there's a process called splicing going to take place. Splicing. So what is splicing? A set of enzymes specific for DNA molecule bind to these regions. The region is it is between this introns and exon region, the interface like here. So between intron and exon, here you see a specific region. So they start to bind this region, this set of enzymes, and they are called a spliceosome. So once a spliceosome bind, they can cut this DNA molecule. So when they cut this DNA molecule, which resulted these pieces, right? So here one piece, and here one piece, and here one piece, and here one, like this. So it is fragmented into different pieces. And the spliceosome at the same time join exons. Let's say this exon is labeled as X, this one is Y, this one is Z. Now these cut exons joined by the spliceosome. So when they join, they can now make a different method. For example, we assume, okay, X is come here, and Y comes here, Z comes here. That's what we expect. But not necessary. They can have only X and Y join. So they can join maybe like this, Z, X and Y, or Y and Z. So here we expect X, Y and Z, but it could be Y and Z, it could be X and Z, it could be Z, Y and X. But now you understand, if they make a different RNA molecule, that's a final RNA molecule. Their codes are different, the orders are different. So this can make a different protein, this can make a different protein, this can make a different protein like that. Now you see, it's a one gene, but it can make many different proteins by the splicing procedure. Okay, we will try to see it again a different way. Right now, you can see exon 1, exon 2 and intron. Only so it shows a small piece with 2 exon and 1 intron. Exons are coding region, introns are not coding region.
RNA splicing removes intron.
Okay, what is going to happen now? So these are the splice site between intron and exon. There are specific areas, splice site. So this splice site, the spliceosome enzyme, they're going to bind as a different complex. Then they can make this loop, and after that they're going to cut this at these two ends. Here it cut from this exon and intron region, and then again they can cut from this region. And they can join these two exons together.
What's the outcome of it when it's done?
Yeah, the significant is, the outcome is one gene can produce many proteins. Because you see the end, there are different sequence of mRNA. Because if we understand for example, let's say the exon region has like A, C, T, G, like different region. So it make a different code at each time. When they make different code, if you can remember genetic code, triplet of nucleotide gives one amino acid, the amino acid order is different. So each of these four pieces, the amino acid order and the number different. After they folding, they make a different proteins. The idea of splicing to make different proteins with one gene. Okay, no need to copy this one because there's this same picture is given in the notes section as a question. Okay, if you see this diagram and the question, exam questions, more than one type of protein can be synthesized from the RNA produced from one gene. The diagram shows part of this process. Here you can see the DNA is unwind and one strand serves as the coding region, and then after that the introns and exons are separated. Then they only make these four regions to make the mature RNA. The mature RNA make this polypeptide chain. Describe how more than one type of protein can be synthesized from the RNA produced from one gene. Use the information in the diagram to support your answer. Now here you have to write the process of RNA splicing. So basically what you have to say, when the DNA is copied to a pre-mRNA, the pre-mRNA molecule has introns and exons. After that, these introns and exons are separated by the spliceosome. So the spliceosome cut these introns and exons from the side, and then they can join only the exons. When they join in these exons, there is a privilege of joining different pieces. Example, because the question says use the information. So you have to use example. In this case, they can make a mature mRNA with BIFG or BIF, like different example you can give. So then based on that, the amino acid sequence are different and order is different. Once they fold into different protein, they can make a different function. Right? That was the question, and then I have given, as this is very important thing that you need to remember well. All the points are given that you need to remember. Right? For example, here first, RNA splicing is a process that removes the non-coding sequence of genes, introns from pre-mRNA and joins the protein coding sequence. RNA splicing is removal of introns and joining exons. That's called a post-transcriptional modification. That means after transcription, post means after transcription. What is the modification take place? The process is called splicing of pre-mRNA. Spliceosomes are used, introns are removed, exons are arranged in different combination. mRNA is different. In translation, different primary structure are formed. So these are the 7 points you need to use to write the answer about the RNA splicing. So different mRNA can produce a different protein. Right, I think there's a space given. Maybe at home you can again write this one in a paragraph form using this point, right? Okay, after that, there is another exam question. Let's do that question. Okay, question is this. A new vaccine has been developed that contain active messenger RNA. The flow diagram shows how this vaccine causes body cells to produce a new protein. First, the active messenger RNA used for the production of a protein is isolated. The active mRNA used to production of protein is isolated. So the mRNA is isolated. Now when you see mRNA is isolated, if a particular cell, whatever cells or whatever structure like this, if they produce messenger RNA and the specific mRNA is isolated. Now how it is isolated? We have this advanced technology. So many advanced technology developed to isolate the specific mRNA. The active mRNA is enclosed in a liposome and injected into patient. Now what is liposome? So you can understand liposome is a structure like this. It is made out of this phospholipid bilayer. Right, so you can see a phospholipid two molecules, and it makes this phospholipid bilayer like this. Right, so they can construct these small tiny balls using phospholipid bilayer. So you can understand the properties of phospholipid, polar head and hydrophobic chain in aqueous space. They always organize into the bilayer. So revise your cell membrane, then you can understand why they make a bilayer in aqueous space. After that, this mRNA inserted into this phospholipid bilayer. So it's called liposome, right? And then this liposome is injected now. When the liposome is injected, liposome can travels all over the body. An advantage of liposome is they can fuse with the cell membrane because they are compatible with cell membrane. This is also a bilayer and the cell membrane is a bilayer. So they can fuse. I think for the last session we have to join again. Okay, then the active mRNA enters the body cell of the patient. So once it injects, let's say this one is a host cell, the person